Sexual reproduction in flowering plants. First is the what is the flower. So we know that in flower there is a filament which is present, which is basically attached to the thalamus, in anther which is bilobe, and bilobe, that is the diathecus, that is the fourth theca, and it is the longitudinal globe. How? For example, what is a bilobe means? This is a bilobe structure, and here are the bilobe means two lobes are present. In two lobes there are the four theca, these are present, so that's why it is called as dithecus. Four theca is present in one lobe. Then we have sauce, which is the female part, that is the stigma, ovary and style, and it is the gynoecium basically. So this is the trick, that is the sauce. Now first is the microsporangium, which is the formation of the microspore. It is a structure in the plants that are reproductive organ where the development of pollen takes place. So microsporangium, where immature stages, it is called microsporangium. This is the outer layer, which is epidermis, and it is the outermost layer where the flat cells are present, and this is the endothecium, which is the radially arranged cells, and then middle layer 3 to 4 parakinematous tissues are present, and then we have tapetum, which is important, which is the outermost layer, and pyramid cells are present and direct contact with the microspores. What is the function of the tapetum? Tapetum provides nourishment, and it produces the hardest known organic substance, which is known as sporopollenin, and it is made up of callose enzyme. Now here it is the theca. Now inside this there is the presence of sporogenous tissue, which give rise to the microspore mother cell meiosis, and their potential cells, which is called the male gametophyte. Then we have microgenesis. Microspore mother cell, which is the meiocyte, and it is 2n in number. They will go meiosis 1, form 2 cells, meiosis 1, and then this is form again. This is a tetrahedral one, it is in outside, and callose, which is the each callose 2 super 8 cells, and then there will be forming the intine and exine. These are the 4 pollen grains which will form, and outer layer is the exine, made up of sporopollenin, which is the hardest known organic substance, and then inner one is the intine, which is made up of the pectin and the cellulose, and this pore is known as germ pore, which is open for the germination. In NCERT it is mentioned that when 60% of angiosperm there is a dehiscence, that 2 cell stage will form. They will form vegetative cell and generative cell, which is the round shape and the spindle shape. And during the 40% dehiscence, the 3 cell stage generative cell divide mitotically, and the viability is the 1 month, which is the solace, and that is the potato basically. And due to the pollen there may be the allergy, which causes the asthma, and there may be these pollen grain can preserve due to the cryopreservation technique, that is the liquid nitrogen, and the temperature is the minus 196 degrees Celsius is the temperature. This is the Megasporangium, that is Megasporangium structure. This is the structure that we have a female part of the plant, stigma, style and ovary, sauce, that is stigma, ovary and the style, and this is the surface landing of pollen, because this is the stigma where the pollen will land, ovary, which is the ovarian cavity, locule, placenta, ovule or microsporangium, and this is the style that connect the ovary and the stigma. Ovary and stigma, they will connect. Then we have the types of pistils. We have single pistil, monocarpillary like the hibiscus, because it is a monopistil. Then we have multicarpillary apocarpus, which is the micalia. This is the micalia. And then we have the multicarpillary like the papaver suncarpus condition. This is the structure of the ovule, that is anatropous ovule. Chalazal and funicle like stalk like structures connecting the placenta and ovule. Now if ovarian have only one ovule like wheat, paddy and mango, and if ovary have many ovules, then papaya, orchid, watermelon are example. Then we have megasporogenesis, like ovule will differentiate, will form this much of the tetrad megasporogenesis, and then they will form, which will form the 8 nucleated stages. After 8 nucleated stages, this is the micropyle and embryo, sac, female gametocyte, antipodal cell, chalazial and central cell with the 2 polar nuclei. They will form 8 cells, 7 cells and 8 nucleated. Then what is the pollination? Transfer of pollen grain to the stigma of the pistil, female gametophyte, embryo sac, male gametophyte, pollen grain, and both are non-motile, that's why pollination. Then we have 4 types: autogamy, geitonogamy and xenogamy. This is the autogamy, xenogamy and geitonogamy. Then these are some of the examples which are also mentioned. We show the true pollination. Clistogamous, cross-pollination, that xenogamy. These are the agents of the pollination during the cross-pollination: abiotic and biotic. Anemophily by wind, then we have corn crab, then entomophily by insect, ornithophily by birds, ceropetrophily by bats, macrophily by snail and slugs, myrenophily by the ants, and this is hydrophily by the water, etc. And reward, which is rich in nectar, lay egg in the pollination. And the 6 feet tallest plant is a Morphophallus. Moth and yucca will lay egg. Moth lay egg, yucca locule, and yucca do pollination. Then we have inbreeding and outbreeding. Inbreeding due to autogamy, self-pollination, no genetic variability causes inbreeding depression, and to avoid this we have to do outbreeding devices to promote the cross-pollination. So outbreeding promote cross-pollination, we do xenogamy. Unisexuality, maize, cucurbita, castor, to avoid the autogamy, and in dioecious plant like papaya, and then we have mulberry, and result is the xenogamy. Then we have self-fertility or incompatibility, diogamy and artificial hybridization, emasculation, removal of anther of stamen from the flower, bagging, the covering the emasculated flower. Then we have pollen pistil interaction. This is a pollen pistil interaction, that fertilization, double fertilization, syngamy and triple fusion, 3 and embryo will form. Then we have the post fertilization event, that is endosperm development, embryo development, ovule maturation and ovary maturation. Remember, ovule mature to the seed and ovary mature to the fruit. Then we have endosperm, which is primary endosperm nuclear, 3N, multiple division and cellular division. Coconut water, white kernel, which is the kernel. Then types: X albuminous and albuminous. X albuminous and albuminous examples are also there. This is the embryo development: zygote, embryonal axis and cotyledons, dicots, monocots. These examples are very important. Then we have seed, which is the fertilized fruit, fruits, true fruits and the false fruits. Like ovary develop into the fruit, true fruit, false fruit like the other than ovary, apple, strawberry, cashew nut. Then we have parthenocarpic fruit, when develop fruit from the unfertilized or without fertilization. Then we have banana, is an induced by growth hormone, cytokinin and auxin. Apomixis, like seed develop without fertilization of ovule, like grass and estriaceae. Then we have polyembryony, many embryo, more than one embryo in the seed, like citrus fruit and the seed, mango, etc. So this is notes of sexual reproduction in flowering plants.